All right, everybody. Thanks for being here. I'm Tim. I'm a graduate student in the McKenzie lab at CU Boulder, PhD student. Broadly, the lab works on disease ecology, but what I'll do today is talk about one of our main projects involving chytrid fungus and boreal toads. And so what I'll do is I'll give a kind of a background introduction to that system. I'll talk about one of our main project from last summer, and then I'll get specific about a few of the things that the Lois Webster Fund supported for our work last summer. I'm not sure how familiar people are with amphibian decline, but it's a massive problem for biodiversity globally. About 40%, now upwards of close to 50% of all amphibian species are threatened, have declining populations. A lot of these are due to the normal threats that all species are facing. Habitat loss being first among them, pollution, invasive species. But there was something unique going on with amphibians that folks noticed around 1980s sometime. They noticed that populations were declining and even disappearing in relatively pristine habitats. Really massive declines, and they couldn't be explained by the typical habitat loss or pollution or invasive species. So we call these enigmatic declines. So Rocky Mountain National Park, obviously a relatively well protected area, saw its boreal toads decline massively. And we didn't really have an explanation for this. There was some evidence that in hindsight looks relatively clear, but back then was not, that these declines were moving in a wave-like fashion. This is an example of when the first declines were recorded throughout Mesoamerica. And it was moving, in hindsight now we can see it was moving in a wave-like fashion, suggesting that a disease was responsible. But we didn't have a disease, we couldn't explain it until 1999. 1999, a lab at the University of Maine first described this new pathogen, and it was a chytrid fungus, and it was found first in this neotropical tree frog, and then later, in the next few years, was discovered to be in almost all the declining populations all over the world. So it was just thought a major threat to biodiversity. And before we had discovered this pathogen, dozens of species had already gone extinct. And it was a chytrid fungus. Chytridium mycota is a basal group of fungus. They diverge really early on in evolutionary history. They are not like the mushroom-producing fungi that you're probably most familiar with, but they do feed on mostly dead organic material, usually in an aquatic environment. They're in that aquatic environment because they have an aquatic zoospore stage. And so there's this zoospore that swims around in the water column and will find a suitable host and infect the skin of that host. A number of amphibian species are susceptible. And once they get into the skin, they'll encyst and they'll grow into a zoosporangia, producing more zoospores to infect skin on the same amphibian or another amphibian that they come in contact with. So this is a picture of an electron microscope image of frog skin with sporangia embedded in there, so the zoospores, once they're released from those sporangia, will swim through the water and are attracted to the chemicals in the mucus on the skin of amphibians and will find a new host. So this is another electron scanning image. And those little round balls there are the zoospores that have just been released. And this is a picture I took on a microscope. Probably can't quite see the zoospores, but they're swimming around those bigger circles which are the zoosporangia. Now this is a really big problem for amphibians because the skin in amphibians is not just a barrier between inside the host and the environment. It's an organ involved in osmoregulation and respiration and excretion and all sorts of functions that are super necessary for maintaining homeostasis. And so when an infection load gets high enough, the host will die. And susceptible species can die within a few weeks. A number of species can tolerate the infection and will not succumb to them, but others will succumb really rapidly. So in Colorado, we have on one end of that spectrum boreal toads, super susceptible, and on the other end we have bullfrogs, which are pretty much fully tolerant. We'll never really see a mortality event from BD. It's also thought that those bullfrogs can be infected and spread the disease to populations that are more susceptible and act as a reservoir for the pathogen. But the thing about amphibian skin is that BD is not the only thing that can live on it. It's a rich mucosal surface. It's got nutrients available. It's relatively moist. And so it harbors hundreds of other microbial taxa, not just the pathogen. This is just a bar chart from a really early study my lab did on the microbial communities associated with amphibian skin. And these are classes of microbes, so a really broad group. And the diversity is really impressive when you look at it. And what's really cool about these microbes that are living on there, largely bacteria, but also other fungi, is that a number of them produce metabolites as they're growing that can inhibit the growth of fungus like BD, the pathogen. So this is an auger plate with a lawn of BD growing on it, and you can see there are two streaks of bacteria growing on it. The one up top is a control one. It's E. coli. It doesn't produce any anti-fungal metabolites. The one on the bottom, the yellow one, is a Lysobacter strain that produces a metabolite that inhibits the growth of the BD pathogen. And you can tell that because there's that clear area around the streak. And so other work in other groups have shown that a higher proportion of the bacteria that produce metabolites like this Lysobacter strain correlate with increased resistance to the BD pathogen. And so the idea that drives a lot of my lab's research at CU is that we can apply living microbes to the skin of amphibians to shift their microbial communities towards one that's more anti-fungal, where a higher proportion of the bacteria on the skin are producing these metabolites that kill the pathogenic fungus. And they don't have any impact on the host themselves, so it's a potential alternative to other chemical treatments. At the zoo, for instance, the most common treatment is an antifungal chemical called itraconazole. And when there's an infection in a captive setting, they're often treated with itraconazole. But that is not super practical to be done in the field on actual wild populations, and B, it's basically a one and done situation. Once you've applied itraconazole and you release them back into the wild, the pathogen is probably still there and they'll be re-exposed. The idea with probiotics is that they are living factories for a antifungal treatment that will persist on the skin and prevent reinfection. And so the main one that we've been using up till now, and I'll talk about some alternatives that were funded by the Lois Webster Fund, is this bacteria called Janthinobacterium lividum, or Jliv. I'll call it Jliv from here on out. It produces this bright purple pigment called violacein, and violacein is strongly anti-fungal and will inhibit BD growth at really low concentrations. So it's a really promising bacteria to use in one of these probiotic treatments in our system. This is a figure from a study that was done in our lab a few years ago. The blue line, and what I can just tell you is that these are in boreal toads, and the blue line represents the percent surviving of a group of boreal toads that received BD and Jliv, the probiotic. You can see about 40% were still surviving at the end of the experiment. That's not great, but when you compare it to the 0% that were surviving in the BD-only treatment, the red line, that's a really significant improvement, and significant enough that might be able to keep boreal toads on the landscape where they would otherwise go extinct. So we've been working with CPW a lot to develop this into a conservation tool to use in the wild. One other thing we noticed when doing this is that the bacterial communities on the skin of captive toads were really, really different from those of wild toads. This is an ordination plot, and I'll show a few more of these later in my talk. But what each point represents is a single, the microbial community on a single toad. And you don't really have to understand the axes, but all you really have to understand is that the closer two points are together, the more similar their microbial communities are, the more shared microbes they have on their skin. And so you see the red dots are all the captive individuals, and the green dots are the wild individuals. And so what this is telling us is that the microbial communities in captivity are really, really different than they are in the wild. And so to use this as a useful conservation tool, we need to be doing experiments, probiotic trials in a wild setting. And so that's what we've been doing with boreal toads. So just a little background on the boreal toad. For those of you who don't know, they're a high elevation species, Colorado's highest elevation amphibian species, up to 11,000 to 12,000 feet. They are, as you can imagine, life up there is really tough. It's snowy most of the year. They're spending over half the year hibernating. They are slow to grow and slow to reproduce, but they are very long-lived. And they, like other toads, in general, they complete metamorphosis in one season. So they'll lay an egg mass right after the snow melts, and then they'll metamorphose in the next few months into fully formed little toads. And so this is a map of the former range of the boreal toad. Most of these sites they've been extirpated from. In Colorado here, we're concerned with the southern Rocky Mountain population that's circled by that oval. It's basically southern Wyoming, all of Colorado and northern New Mexico. There's one population left in southern Wyoming. They've been extirpated from New Mexico. Most of the populations in Colorado have either disappeared or are BD positive and on the decline. And so it's a major priority for Colorado Parks and Wildlife to come up with solutions to keep them on the landscape in the face of this disease, the chytrid fungus. And so now reintroductions are becoming an important tool in that process. So finding a site that's hopefully free of the disease, and then raising some toads in captivity and releasing them into the wild. And so this Project Purple Rain, because our bacteria that we're using are purple, and it started right around when Prince died. My advisor liked that name a lot, so we call it Project Purple Rain. But it's really our attempts to translate those successes we saw in the lab with not curing the BD infection, but limiting the mortality associated with it to a wild setting. And so that CPW at some point in the future could use this as a tool to help maintain boreal toad populations. And so a little background on this year's, or last summer's trial. It took place in Chaffee County in these beaver ponds up one of the drainages west of Buena Vista. It was a really good year for reproduction. Chaffee County is one of the last strongholds of the boreal toad, so we actually had multiple egg masses, so multiple females mating and laying eggs at this site, which is great, but it may be one of the last few in the state doing this. And we collected some of the late-stage tadpoles to try our treatment on. This is what they were looking like in their kind of semi-dried up beaver pond. If you remember, down south there was not a lot of rain or a lot of snowpack last year, or a lot of rain, so it was pretty dried up. We were targeting this life stage for a reason. Previous work we'd done had shown that there was a really strong shift in the microbial community right around metamorphosis, which is represented by that arrow. And we thought that this would be an opportunity for us to seed the skin of the amphibians with a new microbe, in this case a hopefully beneficial one. Just like if a forest fire had come through a forest, it would kind of reset the community and allow other members to become established. That's basically what's happening with the bacteria on the skin of amphibians when they're metamorphosing, when they're going from tadpole to juvenile. And so this is, basically what the setup looked like. It's not super fancy. We had these plastic tubs around the site that we'd store, that were partially submerged with water to put our semi-captive individuals in, and we basically added a bunch of bacteria that we washed off of agar plates, and it represented a small volume, but that volume of bacteria is actually in the order of tens of billions of bacteria, so sufficient to increase the number on their skin quite a bit. And we did this twice within a week. And this is us collecting some of the data that we took. We took really two kinds of data. First off, we do these things called mucosome soaks. Since these are an endangered species, we can't bring them into the lab and subject them to the disease, because at least the controls would most likely die, and we don't really want to subject them to that. So what we do is we soak them in sterile water, and then that water contains the metabolites produced by the bacteria, any host factors, immune factors that leach into the water, and then we take that water and in the lab we challenge it against the pathogen to see what effect it has on the pathogen. And this gives us a measure of how inhibitory the skin environment is to the pathogen. And we also take swabs. So these are the metamorphs. They're very small. The toads get quite a bit bigger, but at this stage they're very small. And we take these swabs of their skin and extract the DNA off them and do what's called 16S marker gene sequencing. And that marker gene sequencing, basically, we will amplify all the DNA on that swab and we'll sequence it, and then we'll get a count of how many of each type of bacteria are on their skin using some fancy bioinformatics tools. Now, so we would say, we have 20% of the bacteria on their skin are bacillus, and 40% are flavobacterium, and the other 40% are Janthinobacterium. And the communities are a lot more complex than that, but that's the kind of data that we get from this sort of approach. And our goal is to increase the proportion of inhibitory bacteria, specifically the Janthinobacterium, on the skin of the frogs, toads, excuse me. We also do mark and recapture efforts. It's really hard to find. I talk about needle in a haystack is, like if the needle had legs, that would be, these are very complex. If you've ever seen a wetland area, imagine trying to find those in a wetland area the size of the campus here. It's not easy. But we do tag them. We do have ongoing recapture efforts with VIE, visible injectable elastomer. It's a little rubbery semi-solid that we inject into their leg. And so this is another one of those ordination plots, some results from this summer from that 16S data. Remember, each point represents the microbial communities found on the skin of a individual toad. And so the closer the points are together, the more similar their microbial communities are. And the blue are the Jliv treatments, the toads that got our probiotic, and the red are the ones that did not. And so if this is day one before anybody got any treatments. You can see they're kind of scattered all over the place. But as we move to a week after the treatment, you can see the red and the blue, the control and the treatment, starting to separate. And then by 2 weeks out, there's a strong separation, indicating that something about our treatment is changing the microbial community. Hopefully, the Janthinobacterium are getting on the skin and clearing the infection from these metamorphs. But we don't know that from just looking at this figure. We did just look at the number of sequences of Jliv that were on them. And you can see the blue Jliv bars were significantly higher than the controls. A significantly higher number of Janthinobacterium sequences ended up on our treated groups than were on the control groups. And this should be enough increase in the probiotic that the skin environment would be inhibitory to the pathogen and would hopefully clear the pathogen. That's some other data we're going to collect this summer, but it's a very promising result. You'll notice there's a giant spread on day 7 in particular after the treatment. And that actually broke down as a result of the stage of metamorphosis they were in. So we were treating around metamorphosis, remember, because we thought this would be the best opportunity to get a new microbe on their skin because the community was resetting. And so here on stage 1 is a really slimy tadpole. Their skin hasn't really started to change to adult-like skin. On the far right is a fully metamorphosed toad, and that transition in the skin is happening here. It's getting less slimy. It's getting keratinized, which is what the BD feeds on. This is a really vulnerable life stage for them because their immune system is suppressed. So this is a really important stage, and so it also, one of the reasons we were targeting this is not just because we thought it would be the opportunity for microbes to get on there, but also because the most important time at which to protect them from the pathogen. And so what you see is that basically the treatment didn't really work on the earlier life stages when their skin was still slimy and not keratinized. The later stages seem to be, as we hypothesize, an opportunity to get our probiotic to stick on their skin and clear the infection. And so, this is a super, super great result, and we're going to follow up a lot with it this summer and try and do more applications. I think, if you're taking anything away from the data that I show you, is which I recognize is a lot. Our application of a probiotic was successful, and that treating their water was a successful way to get it on them. And the success of the treatment is super dependent on the exact life stage that you treat them at, which we're going to look into more as we try and translate into something we can hand off to CPW. What we're going to be doing next year, or this summer, I should say, this field season, is trying some environmental bioaugmentations where we add the probiotic to the soil around their habitat. We will be treating reintroductions. So the CPW has a native aquatics species recovery facility where they mostly raise native fish, but also boreal toads, and those are being reintroduced into the wild. And we are going to do some probiotic treatments on them with the hope that if they're exposed to BD after they're released, that the probiotic will protect them from the BD. We're also looking into other probiotics, and this is what I have to thank you all for, because it is what the Lois Webster Fund supported mostly. We really like Jliv as a probiotic, but We have reason to believe that there are other microbes that live on their skin that might provide better protection. And so we've been, last summer, what we did was survey a bunch of boreal toads, swab their skin, plate them out on agar plates, and isolate those bacteria. And we did that with 131 individuals of various life stages. That's an adult at Zimmerman Lake up north, but those are actually little metamorphs that have just emerged in a site in Gunnison County that we also swabbed. And we've collected about 250 new bacterial isolates for testing against BD, and so far about 11 of them have shown promise as inhibitors that we're going to look into more specifically as potential probiotics. And so, this looks a little, this is what I come back from the field with, from some camping trips with agar plates with a bunch of different bacteria and some fungal colonies on there also. And we take these colonies and then isolate them, and freeze them and do some sequencing on them to figure out what they are and if they're going to be able to inhibit BD. And so that's the work that Audubon supported, and super thankful to everybody who helped contribute to that. I'm excited to share those promising results, and hopefully some of these bacteria prove even more useful to the toads than Jliv. So with that, if anybody has any questions, I'm happy to answer.
Do you have any sense of what caused this fungus to blow up the way it has?
Yeah, it's a super, super important question. And so likely what happened is there were native BD strains in amphibian populations for centuries, and then increased movement of humans and amphibians along with that brought disparate strains together, and they hybridized and produced what it's called is the global panzootic lineage, a pathogen that is now pretty much globally distributed and also hypervirulent, is what we think happened. There's a lot of things we don't understand about it. One of the main things is that the species globally that seem to be the most affected are those that live at high elevations. And we really don't know why. And so there are, boreal toads are part of a species complex, and our ones in Colorado are the ones that live at the highest elevations in that species complex. And the ones that live on the west coast are susceptible to BD, but they don't seem to be, have the massive declines that we've seen in Colorado. And why is that? It's unclear right now. Yeah.
So you had a map of this area where there was red coloring or pink coloring and yellow coloring. Was the yellow coloring the areas where they've been extirpated?
Yes. Yeah.
So there's a lot more yellow area in Colorado than Wyoming. Does that have to do with the elevation difference?
Yeah, it's unclear why the southern Rocky Mountain population is worse affected. Those Wyoming, like the northern Wyoming populations and the Utah population, seem to be less affected, and there's some work going on right now trying to figure out why. It's a super important question, and because you could say, why don't we just bring some of those toads to Colorado, and that's a reasonable question, but in general, wildlife managers are averse to introducing something from another state. But yeah, why those are less affected is unclear. It's not that they aren't affected. There are population declines in other parts of their range. They're just not as severe as they are here.
Yeah. Looks like our population's mostly wiped out.
Mostly wiped out. Yeah, there are 20 breeding populations left in the state. Yeah.
As one of the people who read your proposal a year ago, I want to tell you that you did an excellent job explaining it, because I now understand.
Oh, good. I'm glad to hear that. Yeah, that's good. That's good.
The entire perspective of what you're up to. I just have a quick question. The Jliv that you started with,
yeah.
Is it naturally found on the boreal toad?
Yeah, good question. We the strain that we use was taken off a swab from toad in Chaffee County. Yeah, so they're actually generally a soil bacterium, but they're pretty broadly distributed. They're not in high abundance, but pretty broadly distributed. I have isolates from Chaffee County, from the national park, from Larimer County. Yeah, all over. Yeah. Oh, Mike. Okay.
Will the adults be able to transmit this gel to the next generation, or do you have to re-inoculate?
So it's unclear. There is some evidence of vertical transmission of symbiotic microbes in amphibians in other systems. We haven't looked at that in boreal toads, but people have looked at that in other in tropical amphibians. So it's unclear. My guess is that no, not in enough abundance to make a impact. But how I envision this working as a conservation tool is there are a limited number of breeding sites left and boreal toads in Colorado, and at the last boreal toad meeting we had with all the agencies involved, there was we talk about the status of all the remaining populations, and 3 more of them went positive for BD this year. And so the idea would be go to those sites that are newly positive, treat everybody, and try and get the pathogen load eliminated to keep it BD negative for a little while longer at least. So I wouldn't envision treat a bunch of adults and then in perpetuity their bacterial communities are super inhibitory, but certainly longer, the effect would be longer than an itraconazole treatment or something chemical. Yeah.